And you're live.
Hey everyone, so today's field trip is looking at plankton. And so why do we care about plankton? What's the big deal about plankton anyway? They're important to us because they are the basis of the entire food web for the entire planet. And so without plankton, we are in a lot of trouble. And so plankton are these amazing creatures. They spend their time in the water column and they are moved around the world by ocean currents. They're not strong enough swimmers to overcome the ocean currents, so they're just moved by the ocean currents, and they spend their time in the water column. They move up and down within the water column, but they're just moved around by the water. And so fish is an example of an animal that lives in the water column, but it's not plankton because it is free swimming and it can overcome the currents. So that's the distinction we make. And Maine is in a temperate ocean, and so we have a lot of nutrients in the water here, and so we have a lot of plankton. If you go to the equatorial regions, there's less nutrient in the water and less plankton. So different species of plankton, even though there are plankton in both areas. In the temperate oceans, we have more plankton because we have more nutrients, and in the equatorial parts of the world, we have less nutrients, so less plankton. So Maine and the ocean off of Maine are really productive waters as far as fisheries go because of all the plankton, because our food web is so strong up here. So some of the things that you need to know about plankton, there's some definitions that you need to know. We already said plankton means that they are organisms that are in the water column, and we have phytoplankton and zooplankton. Phytoplankton are the photosynthesizing plankton. They are like the algae or the single cell photosynthesizing organisms, or diatoms are also in the plankton. And zooplankton are the animal portion of the plankton. So they they're consumers of phytoplankton. And another definition you need to know is the difference between holoplankton and meroplankton. Holoplankton, the way I learned to remember it, is they spend their whole lives in the plankton phase. They never settle out of the plankton. They're always up in the water column. So again, holoplankton spend their whole lives in the water column, and meroplankton have part of their life history is that they're either benthic or free swimming, and the other part of their life is up what we call in the plankton. So the larval form of a lot of organisms like barnacles or mussels they're up in the water column. So that is the meroplankton. And I'd like to give you a couple of examples of each just to help you sort this all out. So if we have a phytoplankton, we have an algae, a single cell algae that's a holoplankton. It's a photosynthesizing plankton that spends its whole life in the water column. Those would be the small single cell algae that are always in the water column. And if you want to think about a zooplankton, that would be an animal. So like a barnacle is a zooplankton, and if they spend their whole, the barnacle, if they have a benthic form and a planktonic form, and so they're a zooplankton that's also the meroplankton. Now let's think about a zooplankton that's always in the water column. So a zooplankton that's a holoplankton would be like krill, or some of the other small animals that are they're animals that are always in the water column and moved by the ocean currents. And so those are some examples of the plankton. And you're going to be looking at this on your worksheet as well. So while we're here, we're going to show you how we collect plankton. And we have a couple of plankton nets here, a couple of different sizes. If you want to just do a surface plankton tow, you'd use a small net. And this you would just put in the water and you could just tow it along, and you could see who's living on the surface of the water. Here's a little bit bigger plankton net. I'm sorry, I should have given you the definitions. This part up here is called the bridle.
Can you point to that again?
This part up here is called the bridle and the harness. And you just drop these in the water. if we drop it in the water now, you wouldn't see anything, so it doesn't make any sense to try to do that. And then you tow it through the water, and when you're looking for organisms of a certain size, you put a filter on the bottom of the bag, and you put, in this case, the filter is a 10 micron filter, so any organisms that are smaller than the 10 micron size will be filtered out. And so you put this on. This is pretty heavy, so when you drop it in the water, it'll sink down, and then you just tow along, or you can do a depth. You can drop it to a certain depth and tow up, and you can get a sample of the water column, any organisms, in this case, less than the 10 micron size. All right, and then if you wanted to tow behind a boat, and this is all down at the Schiller Coastal Study Center, if anybody wants to in their time at Bowdoin study plankton. This net you can drop overboard. You put the same filters on the bottom, on the cot end of the bag, and you can keep track of the depth of this by the radar that's on the boat. Captain Paul Joyce, who runs the boat, he works well with us when we have to go out. So you could sample at the surface, you could sample at 10 meters, you could sample at 60 meters, and he will keep track of the depth, and you can keep track of the length of time that you want to do your tow. It gives you an idea of how throughout the seasons the plankton change, their distribution and their density. And another way to check density is with a thing called a Secchi disc, and I am now going to take the camera, and Justin is going to show you how to use the Secchi disc.
Okay.
All
cool.
right.
Go for it.
Yes.
All right. So we spoke earlier in the semester, and I guess earlier in this video, about how plankton are collected, and that we have really productive waters here off the coast of Maine. So one way that we can measure productivity, or what this is called a proxy, is we can measure essentially the amount of depth down that we can still visibly see, so that visible light passes, and that is the photic zone. The photic zone is where a lot of our phytoplankton are going to be living most of their lives, because they need photosynthesis to get energy, right? They need sunlight. And our zooplankton are going to move up and down in their water column in order to feed, and we're going to get into that in lecture. So one way that you can measure depth, or the depth of the photic zone, is with using this thing called a Secchi disc. And it is the most sophisticated piece of science equipment you could ever imagine. It's literally a piece of fiberglass with two colors on it. So the two colors are white and black as a matter of contrast. So we can drop this guy over the edge of the water, and all we do is we drop it down until we can't see it anymore. So we take a nice measurement, we drop it down, and we wait for it to disappear. And when you can't see it with your naked eye anymore, you bring it back up a little bit until you can see it again. And then that depth at which you can no longer see it is the Secchi depth, or the proxy for the depth of the photic zone. And that tells us how productive the waters are, what kind of nutrients and sediment we might have, and it also gives us an idea of kind of where we would be looking for our phytoplankton. So we could then set our phytoplankton tow to only tow through the photic zone, because why would they be below that? So that's how Secchi discs work, and usually when we're collecting plankton or fish tows, we want to get an idea of depth of the photic zone, so we use this in addition to that. That's it for us.